Bismillahirrahmanirrahim. Our topic for today is it's a gram-negative rod. So we know all gram-negative rods are pink upon gram stain. Now, the particular feature about this organism is that it consists upon specific antigens which help in its identification and its serological typing. Now, the first basic one is the O antigen, which is present on the cell membrane. Now, this is a polysaccharide which helps it to divide into multiple groups, which are from A to 1. Now our next antigen is present in this capsule, which is also provides its phagocytic action, which prevents it from being eaten. Now this antigen is VI antigen, or so I call it like that because I read it as that. So this is also used in clinical diagnosis. And last is the H antigen, which is found in the flagellum. Now there is not multiple types of these antigens. There are only two forms depending upon the gene sequence in which the M messenger RNA is aligned at that time. So if the messenger RNA is aligned in such a feature, irrespective, this is not quite important. So basically you need to know is there are two forms, and either form can be present, but they're particularly to one. Now we'll go next towards the classification. There's many classification. The first one being the Ewing classification. Now, the Ewing young man, what he says is that he divided the organism into three genuses, such as the S. typhi, the S. cholerae, and the S. enterocolitis. Now, he said that the first species consists upon only one type of serotype. Similar was with the cholerae, but the third organism, S. enterocolitis, consisted upon 1500 serotypes. Now another classification came in after some time, which was known as the Kaufman and White assignment. Now this person assigned different species names in each serotype. Now he gave about 1500 names and formed 1500 Salmonella type of species. Last classification is DNA hybridization. In this, what happens is the S. typhi is not a distinct species which is classified in the S and given a special species name. It is under a serotype S. enteritica. S. enteritica. Now, our latest classification, which we normally follow, is the clinical classification. Now, according to the clinical classification, the organism is divided into two groups. Either the organism will cause typhoid, so it is known as typhoidal organism, or typhoid fever causing, or non-typhoid causing organism, which includes the enterocolitis, the metastatic infections such as osteomyelitis, and etc. This is quite simple. That this organism causes multiple type of diseases that can cause diarrhea, in UTI, and even neonatal meningitis. But we will break it down into three bigger diseases. The first one being enterocolitis, which is exhibited as diarrhea, septicemia, and typhoid. Now we make a triangle in order to simplify it. Okay, now what happens when this organism comes? But before knowing that, you need to know is that this organism is normally flora in the large intestine, so means it is abundantly present over there. So whatever will happen is that if it ascends or enters anywhere apart from its normal home, which is the large intestine, it will result in infection. So whenever we acquire this organism by orophycal route, so this is by handling of the people who handle your food, and if it is not properly good, this organism will enter. But in order to cause its infectivity, a large amount is supposedly to enter your body. Now, we know large intestine is not affected by it because it is already present there. Why it is present? It is not affected by it because the endotoxin produced by this organism does not have receptors in the large intestine. So, large intestine remain unaffected. So, whenever this organism enters the small intestine in large amount of quantity, this will result in an infection. The first infection we will study is enterocolitis. Now, this organism enters And what it does is it penetrates the mucosa and the epithelium and resides in the lamina propria. Now you know lamina propria is abundant in neutrophils. Now whenever this organism enters, there will be a neutrophilic reaction and there will be inflammation. Now because of such phenomena, there will be a lot of inflammation, there will be leakage of fluid from these blood vessels. Hence the patient would have symptoms like pain, nausea, abdominal pain, and diarrhea. The diarrhea can be either very mild to severe, and it can be associated with or without blood. Now, the amount of blood which is present is very, very less in quantity, but it is present in such an organism. Now, this type of diarrhea affects mostly, this is mostly self-limiting, but it can be severe in very young or either very old patients or patients who are HIV positive, which meaning that if they have a low CD4 count, they will have severe symptoms, and there will be a probability of this organism metastasizing into the blood, resulting in bacteremia. Now, the other disease which we are going to study, which is caused by this respective organism, is the typhoid fever, which is a common condition. Now, in this, you have similar organism entered your body, but this time the species was different. This was a typhoidal species, as we have discussed in the previous classification. So, the typhoid species, what it does is it enters the intestine, but this organism, if enters the small intestine and where the pear patches are caused. Some of this cause diarrheal symptoms, but some of them enter the pear patches. In the pear patches, what is present is there are a lot of macrophages. Now the macrophages are bacteria engulfing cells, as you can see from this macrophage mean look. Now this macrophage, when engulfs such an organism, they do not die inside it. Instead, they start multiplying. As you know, all of this system is quite connected. From this system, these macrophages ascend upwards by the blood into the liver, into the gallbladder, and even into the spleen. So if this organism enters such a respective area, the patient becomes chronic carriers, and they reside in such organism for a long period of time, and their survival is maximum because of they are able to survive in these phagosomes. Now, what would be the symptoms manifested by in such a disease? Now, typhoid species are of two types. They are S. typhi and S. paratyphi. S. paratyphi is further divided into A, B, and C. A consists upon S. quatermulari and B. Hirschfeldi. Now, the onset of this typhoid is quite slow. It is also associated with a lot of few diarrheal symptoms. But in the starting of this disease, what happens is the patient starts on with a fever and constipation. In the first week, they do not know what is going on until they reach the second week. In the second week, there is a step ladder fever, which slowly increases, and there is delirium over time. There is tended abdomen, and the patient develops slight enlargement of spleen. We will discuss the clinical features again after some time. So, moving towards our last disease, which is actually septicemia. Now, septicemia, as you know that the organism has crossed the intestine and it has entered the blood, and it results in bacteremia. Now, in this, it does not affect a lot of patients. It only affects two types of people. It can occur in two types of settings. Now, one, in those patients who have chronic illnesses. They are chronically ill. Chronically ill meaning that they have genetic abnormalities such as sickle cell anemia, in which they are in constant state of crisis, or they have cancer. Other setting that can appear is in children. Small kids are suffering from enterocolitis. Now, if they are suffering from enterocolitis for quite some time, approximately into 5 to 10% of the patients, this disease will be affected. Now, now if in such a patient, what happens is they have the tendency to enter the blood, and similarly from the blood they can enter different locations, such as they can cause osteomyelitis in the bones and even pneumonia. They can affect the lungs and resulting in pneumonia. Enterocolitis is that the patient will have lots of nausea accompanied with vomit, abdominal pain, and diarrhea. As we know, this specific enterocolitis is mostly caused in the US by a specific serotype which is known as S. typhimurium, as from an epidemic which was caused in the earlier 90s in the US, in which there was a maid who used to cook food for a lot of people. Because of that, maid carried such a serotype. They were all of their hosts were affected, and since because of massive concentration was affected, hence this species was given its specific name, clinical feature, which is present in typhoid. Now, the causative organism in this bacteria, this genus is S. typhi and S. paratyphi. Now, this disease has a very slow onset, and it starts with a fever and constipation in the first week. In that time, patient is very unknown of what's happening. In the second week, what happens is there is ascending type of fever, which is a very high grade fever, which has no intermittent value. Patient can also be accompanied with delirium, a very tender abdomen, and slight splenomegaly. Rarely what happens on the abdomens are there are reddish macules, which are known as rose spots. In the laboratory finding, what we can see is there would be anemia, as in you can see the HB would be they would be giving a picture of a less amount of RBCs or anemia type of picture, and there will be decrease in blood vessel white blood cells, meaning leukopenia, and in some rare cases there are even deranged LFTs. Now, if the disease continues after the second week, the patient has entered a danger state, which is a complication. It is a complication week. So in this stage, what happens is there will be an intestinal perforation or even intestinal hemorrhage. Approximately 3% of the people can be affected by this, and in those 3%, what are those patients are also carriers, because what happens, we have previously discussed that from the intestine, from the pear patches, the phagosomes ascend upwards towards the liver, affects the liver, the gallbladder, and the spleen. So if its concentration even after disease is present, so these people become chronic carriers, and they excrete Salmonella typhi for a prolonged period of time. Hence, because of this, there are other people have more tendency to be affected by such people. Now, always know the organism itself can never be acquired because it resides in the phagosome, but the toxin can be found in the stool sample. Now, lastly, we go towards the organism as cholera suis. Now, in this, what happens is the organism has sufficiently entered from the intestine into the blood, resulting in bacteremia. From the blood, it goes mostly to the brain, resulting in meningitis, lungs, resulting in pneumonia, and even bones, resulting in osteomyelitis. enterocolitis is quite simple. It is a self-limiting disease, so it will immediately stop on its own. But what you need to remember is that the incubation period of such organism is about approximately 48 hours. So you need to differentiate it from other bacterial organisms, resulting in different types of infections. So treatment should be according to what type of empirical therapy you are going to give. So the first empirical therapy is giving the patient approximate amount of fluid and electrolyte balance, because in diarrheal state what happens is there is a lot of dehydration, so we have to replace that fluid with more fluid. Now we can also use antibiotics. What it does is it does not shorten the illness or reduce the symptoms, but it inhibits the prolonged excretion of the organism. And lastly, we can also give antimicrobial drugs. This is only recommended in neonates because the severity of disease is quite more. Now, we'll discuss what we do in typhoid and septicemia. Now, typhoid and septicemia is a very, very critical condition. This type of infections is ceftriaxone. Exone. Another drugs which we can use is ciprofloxacin. But in chronic carriers, the first drug, particularly chronic carriers such as species affected by S. typhi, is ampicillin. Next is ciprofloxacin. Chronic carriers are also recommended to undergo cholecystectomy in order to remove the gallbladder which is infested with S. typhi, because later in life it can also result in focal abscesses and other problems such as osteomyelitis because of this organism inhibiting such a human body. Our identification of this organism is that we grow this organism onto an McConkey agar and EMB agar. EMB and McConkey. Because this is a rod, and after gram staining it, we need to know that if it is a lactose fermenter or not. So there is no fermentation seen. Hence, we derived a conclusion that this is a non-lactose fermenter. Hence, we move towards our next test, which is confirming if this organism produces H2S gas or not in order to identify this specific species. Because whenever you see a gram-negative rod and the patient presented to you with symptoms such as diarrhea, you need to know if it is Salmonella, Shigella, or etc. Now, there are many tests. The first test in lactose ferment is the lactose, checking if the patient organism is lactose fermented or not, which we previously did, that this organism is not lactose fermented. If it was a lactose fermented, it would probably be E. coli. But since it's not, we move towards next test, that what is it? So our next test being the TSI test, which helps in confirmation of this organism. And this, what happens is, we see an alkaline slant, which we will see in by this, and we have an acidic part. But the particular feature of TSI in confirming that this organism is Salmonella, that it has H2S gas origination. Now this helps us in identifying the organism, unlike Proteus, which has similar effect. But the main thing about this is that this organism is urease negative, whereas Proteus is urease positive. Similarly, other organisms like Shigella, Campylobacter, and H. pylori can be ruled out by using such a test. Now, other things which we can also use is by doing blood cultures. Now, blood cultures identify the organism in the first 2 weeks of this disease. We can also use a bone marrow culture, which also turns positive. And lastly, we can also culture a stool, but because of the lot of cross-reactivity, it is mostly positive even in chronic carriers. Lastly, in epidemic conditions, what we can use is for quick identification of this organism, and irrespective of what serological even type is present, we use a Vidal test. Now this is not this is used in certain areas, as I've told you, but this test is not able to identify the organism. Instead, it identifies the antibody titer, which is specific to the antigens present on the cell wall, cell membrane, or the capsule, or even the flagella. So this test is respectively in order to identify the concentration of this organism is a lot in that respective body. In such patients only this test is possible. This does not have a particular vital use in laboratories, because in most people it turns out positive, but if it turns out in a larger amount, and it is used in basically epidemics.